guys, welcome back to ASA HUB. This is Shama, and today I'm here with you guys with an interesting topic, gene editing using CRISPR-Cas9. We came up with this topic because some of our subscribers has requested for this one. First of all, you should know what is gene editing. Gene editing or genome editing is a group of technologies that gives scientists the ability to change an organism's DNA. That is, if we want to alter an organism's DNA in order to get a result, I mean, if we want to alter, for example, like quality of the crops or nature of the fruit or nutrients of the fruit should be rich in something special. So for that, we can do gene editing. And gene editing is a technique in which we can add the genetic materials, remove or alter them at particular locations in the genome. So, what is genome? Genome is an entire genetic material present in an organism. So, in this modern world, there are so many approaches for genome editing, and one of the well-known and the best is CRISPR-Cas9, which we are going to learn. So, CRISPR-Cas9 is a short for clustered regularly interspaced short palindromic repeats, and Cas9 stands for CRISPR-associated protein 9. CRISPR-Cas9 system has created a lot of excitement because it is faster, cheaper, more accurate, and more efficient when compared to other genome editing methods. So, when coming to CRISPR-Cas9, it's a unique technology which enables the medical researchers to edit the part of the genome by removing, adding, or altering the sections of DNA sequence, as we have already discussed. So, if you want to alter, or if you want to alter any product or result of a particular organism, we would edit the DNA. So, in order to attain the editing, or in order to achieve that, we use this CRISPR-Cas9 technology. So it was actually adapted from a naturally occurring genome editing system that was actually a part of immune defense of bacteria. So in short, when we say when a bacteria gets infected with a virus, the bacteria, we know that the virus injects their DNA or their genetic material into the host organism. So here the bacteria capture the small pieces of virus DNA and they insert them into their own DNA and form a particular pattern to create segments known as CRISPR arrays. That is, clusters regularly interspaced short palindromic repeats. So there will be many CRISPR arrays in different parts of the DNA segments, and this allows the bacteria to remember the viruses. I mean, closely related viruses. That is, after a long time, when the viruses infects the bacteria again, they remember that actually they have already been infected with such a virus, and what should they do if the virus attack again? The bacteria produces RNA segments. So this is the most important idea behind this. So here they create the CRISPR arrays, and then if the virus attack again, bacteria produces an RNA segment from the CRISPR array, and that is able to recognize and attach to the specific region of the virus's DNA. And this attachment, this RNA segment leads to the attachment to the specific region of virus DNA, and the bacteria uses Cas9. Cas9 is actually a protein used to cut the DNA apart, and this gets attached to this segment, RNA segment, and this enables the cutting of DNA of the viruses actually, which actually disables the viruses. So this is the main mechanism, or this is the main application that means this is the main principle behind CRISPR-Cas9. So we have discussed what is the principle behind CRISPR-Cas9 system, and researchers have adapted this immune defense system to edit the DNA, and they create a small. So here, what do they do? Is that they create a small pieces of RNA with a short guide sequence. This is called as guide sequence that attaches binds to a specific target sequence in a cell's DNA, and the RNA segment bacteria as the RNA segment bacteria produces from the CRISPR arrays. So here the meaning is that In natural adaptation or naturally in bacteria, when the virus infects the bacteria, it produces a CRISPR array, and from that, it produces an RNA segment. But in actual, I mean, in research field, what the researchers do is that they create a small piece of RNA, and it is called as guide sequence, and this attaches to the specific target sequence in cell's DNA in order to edit that part. So it works in similar with the RNA segment produced from the CRISPR array. And this guide RNA is also able to attach to the Cas9 enzyme that we have already discussed. It cuts the DNA apart. And when introduced into the cell, the guide RNA recognizes the intended DNA sequence, and Cas9 enzyme cuts the DNA at the targeted location, mirroring the process in bacteria. It works as similar in the bacteria, only the difference is that we introduce the RNA, which is called as the guide sequence, in order to edit the gene. The Cas9 is the enzyme. I mean, actually, most often Cas9 is the enzyme that is used, but other enzymes such as CPF1 is also used. Once the DNA is cut, researchers use the cell's own DNA repair machinery to add, delete pieces of genetic materials or to make changes to the DNA by replacing with customized DNA sequence. It is currently the simplest, most versatile, and precise method of genetic manipulation, and thus it has been causing a buzz in the scientific world because more and more genetic changes or genetic editing can be done from this current editing mechanism. When coming to more details of CRISPR-Cas system, it consists of mainly two key molecules, that is enzyme and the RNA, which is called as the guide RNA. So these two key molecules are used to introduce a change in the DNA. So a change in the DNA is known as mutations, and the enzyme Cas9 is one of the key molecules. This acts as a pair of molecular scissors. As we have already told that it is used to make cuts in the DNA strand. Okay, so at specific locations. So we use Cas9 as a molecular scissors so that it can cut the two strands of the DNA at specific locations, and by that we can add or remove the parts of the DNA. And the other important molecule is the RNA or the guide RNA, also known as gRNA. This helps to guide the Cas9 enzyme towards the DNA where the cut is to be made. So this RNA consists of a pre-designed RNA sequence. Actually, this pre-designed RNA sequence is always complementary to the DNA where we have to make the cut, and a longer RNA scaffold. The scaffold part binds to the DNA, and the pre-designed sequence guides the Cas9. So the scaffold means the guiding part of the RNA guides the Cas9 to the right part of the genome, while the scaffold part, it has also a longest RNA scaffold. So the scaffold part binds to the DNA where we have to make the cut. So this makes sure that the Cas9 enzyme cuts at the right point in the genome. Then the guide RNA is designed to find, and as we have already talked, the guide RNA is designed to find and bind. So this term, find and bind to a specific sequence in the DNA, and the guide RNA has RNA bases that are complementary to those of DNA sequences in the genome. This means that the guide RNA will only bind to the target sequence and no other genome of the genome. So by making sure that it does not bind to other genome or other parts of the region of the genome, we can edit the DNA as we need. Okay, so the Cas9 follows the guide RNA to the same location because the Cas9 is led by the guide RNA. So where the guide RNA binds or where the guide RNA binds to the DNA sequence, the Cas9 also will follow the guide RNA and makes a cut across the both strands of DNA. And at this stage the cell, because in DNA if there is any error in between, the cell will recognize the damage and it will try to repair it. So during the repair mechanism, the scientists can introduce the changes to one or more genes in the genome of the cell of interest. So by this way the gene editing is done. So here is the diagrammatic representation where we can see that the DNA is here, and this is our target sequence where we have to edit or where we have to introduce a particular bases or changes. So here, when the guide RNA is sent, it will bind to the complementary part. Actually, this guide RNA is made complementary to the target site, so it will directly bind to here only. No other regions is complementary to this guide RNA, so it only bind here. And after binding, the scaffold part binds to the DNA, and the guide region or the guiding region binds to Cas9, because the Cas9 enzyme binds to the guide RNA region. So the Cas9 will come and bind to that part, and then it will make a cut. The Cas9 enzyme cuts both strands of the DNA, and this cut, when is repaired by introducing mutation. That is, when the cell undergoes repair mechanism, it will eventually complete or repair this cut, so leading to a mutation. So these are the basics of CRISPR-Cas9 in gene editing. So if you understood, or if you found it easy to understand, please like, share, and subscribe our YouTube channel. Thank you.